Bismillahirrahmanirrahim. Dear honorable speaker, special guest, research enthusiast, participant, good morning. Welcome to your most expected research idea series. I'm MD Arju Hossain, Campus Research Ambassador of গবেষক হতে চাই, B Researcher BD, platform from Mawlana Bhashani Science and Technology University. It's my pleasure to anchor today's program. Before dive into the lecture, I will introduce গবেষক হতে চাই, BE Researcher BD platform, its mission, vision, and activities. গবেষক হতে চাই, B Researcher BD is an online-based, non-profit open platform directed by some world-class scholars. They are tirelessly working to promote research and guide young researchers in Bangladesh. Its vision is to leverage research in higher educational institutes for sustainable development all over the world. This novel effort has been initiated by Mr. N.D. Sabir Hossain sir. He is working as an assistant professor at the Department of Computer Science and Engineering of Chittagong University of Engineering and Technology, Kuwait, Bangladesh. Universities are the place for knowledge creation and dissemination. However, we know that the universities are going in the traditional way. Although the finalist students do their thesis or project for obtaining this certificate, but research and publication at undergraduate level are rare. Mr. Sabir Hossain sir work in a different way. By his efforts, the secondary students of the CSE Department of Kuwait have published more than 10 articles in international conferences and journals. This type of accomplishment is rare, not only in Bangladesh, but also in first world universities. Being perplexed by this achievement, he has created this platform to diverse this enthusiasm among all of the students who are interested in higher studies. They are assisting research by sharing research-related resources such as books, papers, websites, etc. Conducting online, offline training, arranging seminar, workshop, or webinar, research collaboration, arranging internship opportunities. Previously, they have conducted the country's first pre-complete online training on how to become a researcher. This course covers the basic concept, tools, techniques required to become a researcher from zero knowledge. The recorded lecture on YouTube received more than 26,000 views within 8 months only. Then they have started the most demanding online free course titled Python for Research from an discipline on 31 April 2021. We know that selecting research idea is the most important step in research methodology. However, finding a research topic is a challenging job for the research enthusiast new learner. To assist them in finding ideas, usually we are organizing this research idea series. Research scholars from home and abroad of different disciplines will deliver lecture related to his research expertise or domain knowledge. Basically, speakers will share trending research topics, skills, tools, and techniques required to start his current and first research project publication guidelines for a specific field. We have a plan to cover all possible research areas in future. Now I am going to introduce today's lecture and speaker. It is the 7th lecture of the Research Idea series. The title, Advancing Epigenetic Therapy for Untreatable Cancer. The key highlights of this lecture are recent advancement of cancer therapy, beating cancer with epigenetic inhibitors, skills, tools, and techniques required to start, speaker's current and first research project, publication tips, and research collaboration. Today's honorable speaker is Dr. Abdullah Al Emran sir, who is currently working as a postdoctoral researcher at the Melanoma program of the Centenary Institute. He also holds a research fellow position at the University of Sydney. Dr. Emran has completed his PhD in cancer biology and epigenetics from the University of Queensland. Previously, he received his BSc and MSc from the Department of Genetic Engineering and Biotechnology of the University of Dhaka. Dr. Emran sir then joined the Department of Biotechnology and Genetic Engineering of Mawlana Bhashani Science and Technology University and served as an assistant professor and also head of the department before pursuing his PhD in Australia. He is fully motivated to train and educate the next generation of enthusiast scientists in biotechnology, biochemistry, molecular and cell biology area. His research interests are in cancer epigenetics, biological science, adaptive resistance system, translational oncology, immunology, therapy, and bioinformatics. The lecture will start now. There will be a question answering session after the lecture. You can ask questions by commenting on this live program on our page. Now I am requesting our honorable speaker to start today's lecture. Thank you, sir.
Thank you very much, Arju, for your nice introduction. And thank you, BD researcher and Gobeshak Hote Chai, Bangladesh, for inviting me for this seminar. Without any delay, I'd like to start my presentation. The topic has been already announced. It's advancing epigenetic therapy for untreatable cancers. So my presentation will cover the following topics, namely as research outcome. So I'll briefly talk about my research outcome, and I'll start with discussing some recent advancement of cancer therapy, and then I'll discuss about the prospect of epigenetic therapy, and then I'd like to discuss few tips for skill sets, publications, and research collaboration. So what is the recent advancement of cancer therapy? So if we look at cancer at a glance around the world, it is the second leading causes of death worldwide. The numbers are really staggering. If we see the global cancer incidence, it is like 18.1 million new cancer cases each year around the globe. And in terms of mortality, I can see there 9.6 million cancer deaths occur each year. And if you look at to different continent, Asia has the highest number of cancer death in each year. So as you know, what is cancer? This is uncontrollable form of cell growth, and if it is detected early, it is better. But if it is not detected at the early stage, which we call the primary stage, then it is metastasized. That means it transforms into different parts of the body, and where it is really uncontrollable at the metastatic stage. So if the cancer cannot be detected at the early stage, which most of the cancer cases does, like it is mainly detected at the very late stage, then it is problematic to provide the correct therapy, although there are different therapies available at the moment, and that causes mortality. If we see the brief history of cancer therapy, first it started with the surgical treatments, and then there was advancement of radiotherapy, and there are a lot of advancement with the chemotherapy. And then in the 1980s, targeted therapy has introduced, where cancer patients were treated with different tyrosine kinase inhibitors. And then the latest, over a decade ago, there has been introduction of checkpoint inhibitors or immunotherapy. And that has led to a lot of progression of cancer therapy, particularly in skin cancer like melanoma, as well as in lung cancer. With the advent of immunotherapy, there has been a lot of success in patient survival. However, there is rapid emergence of resistance develop in the patient, which I'll talk later. For example, as you can see in melanoma, um, after introduction of chemotherapy as well as interleukin therapy, there has been a lot of progress with targeted therapy after 2010, and around 2013 or 14, there has been introduction of immunotherapy, which has revolutionized actually the therapeutic prospect of skin cancer, particularly in melanoma. But I'll discuss more, as I said, in terms of what is the problem with the current therapies. So the next question comes that why we would like to have alternative therapy like epigenetic therapy, and the question may arise that whether we can beat cancer with epigenetic therapy. I'd like to address few findings of my PhD and postdoctoral research from now on, and then try to answer some of the questions that we have raised now. So what is adaptive resistance or acquired resistance? I'm talking about adaptive resistance or acquired resistance during the cancer progression. So there has been a lot of studies, extensive studies done so far with the mechanism of adaptive resistance. As you can see here, there has been a lot of study previously shown that somatic mutation, drug inactivation, alteration of target, increase of signaling pathways, and various other mechanisms can cause adaptive resistance. But recently, just over a decade ago, there is a growing appreciation of understanding the role of epigenetic mechanism of adaptive resistance. So where researchers around the globe have found that adaptive resistance can occur without any genetic mutation, which suggests an alternative mechanism like epigenetic mechanism. I'll talk about the epigenetic mechanism a bit more in the following slides. So what we have done previously in the lab, that we try to understand what happens during the early phases of adaptive resistance. So what we called, we found that transient acute resistance develop as a generic response, and that the cells that survives the initial stress-induced response called induced drug tolerant cells, or IDTCs. This is a sad example of a melanoma patient that we often provide, where you can see that before the treatment, he had melanoma actually everywhere. So it is like metastasis patient. And after introduction of a targeted therapy, which is called BRAF inhibitor, there has been complete remission of cancer, and it was like amazing. Clinicians were amazed with the results they have got, and patients were also very pleased with the response they have received. But unfortunately, after a few weeks, there has been development of resistance, and the patient unfortunately died after 2 weeks. So around 25 to 28 weeks, the patient died. So previously, we have tried to understand that what happens during this phase that leads to the transition to this more aggressive form of tumor, and that caused relapse and ultimately death of the cancer patient. So we have identified different phases of adaptive resistance, namely early IDTCs, late IDTCs, and that ultimately progress to permanent resistance. So this phase actually after 2 to 3 weeks of drug treatment, and this is after 4 weeks of drug treatment, 4 to 6 weeks of drug treatment. That means around 30 to 40 days. And this has been developed after 3 months of drug treatment, which is like around 90 to 120 days. So we have hypothesized that different cancer may share common feature during the early adaptive resistance stage, and this might be happen due to epigenetic remodeling or epigenetic reprogramming, and that might cause permanent resistance or adaptive drug resistance. So for my PhD, I actually focused on this first part, which is early adaptive resistance. And I have, so the basic idea was that if we prevent the formation of this early adaptive resistance, we can ultimately prevent the formation of permanent resistance, which is not responsive to any drugs, actually. So it is like 100 or 500 times resistance to the therapy that initially introduced. So I basically tried to characterize this phase called early IDTCs, and I tried to understand what is the mechanism of this transition, and I tried to understand the functional validation. So for example, if I identified that these are the main mediators of early adaptive resistance, can I prevent that by genetic manipulation or with any drug? So basically, I have tried with various cancer cell lines representative of either melanoma, lung cancer, colon cancer, breast cancer, or liver cancer. And I have treated them with either chemotherapy or targeted therapy, as you can see here. And I have tested various concentration to optimize the optimum doses of IDTCs. When I generate these induced drug tolerance cells or early IDTCs across different cancer cell lines representative of different cancer types, I found that they become cost resistant to high concentration of chemotherapy or targeted therapy. And interestingly, they become growth arrested soon after we started the treatment. As you can see here, after 12 hours of drug treatment, they become growth arrested, which is represented by the red color, and this green color represented that means they are in the cycling phase. And they maintain this growth arrested state after generation of IDTCs for 12 days. So what we have observed that, sorry, there was a hyperactivation of different pro-survival signaling pathways that are essential for cancer progression, as shown in here, that main pathway like MAPK and PI3K pathway, those are essential for cancer cell survival. Those were upregulated once we treated them with drugs or any other stress condition across different cancer types. And we also found that they are also become flattened, translucent. I'm not showing here due to the time limitation. So that led to the research question that what could be the mechanism for this early adaptive resistance. So now I want to introduce the epigenetic modification. So epigenetic modification is the changes of gene expression without alteration of DNA sequence. Um, that means without any mutation, the expression can be changed. So expression of a gene in terms of biology, we say that it may change the protein formation, mRNA formation. So that means without changes of any DNA sequence, the ultimate signal, that means the protein formation, can be changed. This is what we call the epigenetic modification. So this is a nice example of how we show the epigenetic modifications in terms of in a simpler way. So if we look into the cell and if we look into the chromosome, which actually encodes the biological information. In chromosome consists of chromatin, which you can see here, and it is like a bowl of spaghetti or noodles, whatever you want to say. And then chromatin, if we go further down or further smaller structure of chromatin. So chromatin is consist of DNA, and inside DNA there are regions which actually encodes biological information, that is called gene. So DNA or gene is around actually wrapped with histones. These are proteins that actually contains the DNA or chromatin regions, and when it is tightly packed, as you can see here, there will be no gene expression. That means RNA polymerase or transcription factor cannot bind to this tightly wrapped gene or DNA sequence around the histones. But when it is relaxed, as you can see here, that means more open formation of chromatin, then RNA polymerase or transcription factor can bind to the DNA, and then it can transcribe to RNA and then protein. So what actually happens that, as I said, that epigenetic modifications, there could be either modifications of a chemical group onto the DNA. There could be modifications on the histone tails, and so on. So I'll go into more details in the next slide, particularly on to the histone modifications, and why we were interested about histone modifications in details. So as I said, that histones histones are proteins which actually contains the DNA sequence, and in a simpler way, I would say it contains the genes. So if the histones is tightly packed, that means it is repressive. If the histones is relaxed, that means it is active. So there are different modifiers actually lay on different chemical modifications. There are writers which actually write a particular modifications onto the histone's tail, like phosphorylation, methylation, acetylation. There are readers which actually recognize these modifications, and there are erasers which actually removes these modifications. So depending on the type of signals they get from the environment and they get from the cell, this could be either activatory or repressive. And when there are alteration in the histone modifications, that means whether there is more upregulation or more downregulation than the normal cellular condition, then when it occurs disease condition. That means when there is more expression, higher expression or downregulation of these histone modifiers, it causes cancer and other diseases. We were particularly interested about three distinct histone modification on the histone 3 tail, namely HTK4-ME3, HTK9-ME3, and HTK27-ME3, as because the main modifiers that are responsible for these three distinct histone mark were upregulated in our from our gene expression data. And what we have observed that during the early phases of adaptive resistance, there was downregulation of HTK4-ME3 mark and downregulation of HTK27-ME3 mark during the formation of RETCs, and there was upregulation of HTK9-ME3 mark. It is independent, so what we have observed, it is not only for a particular cancer type. We have found that it is more generic. It is like generic for any cancer types or any stress condition. As you can see, I have tried with other stress condition like nutrient starvation. We've also tried with hypoxic condition as well, and what we have observed that there was similar alteration of the histone modification. And when we tried on the in vivo model, we have also found there was increase of this repressive histone mark, which is HTK9-ME3 during the early phases of adaptive resistance. We wanted to know what are the molecular changes during this early adaptive resistance state, and we have found that there are a lot of enrichment of the interferon signaling pathways during the early adaptive resistance. And it has been shown previously that chemotherapy or radiotherapy can cause hyperactivation of interferon signaling inside the cancer cells, which actually helps to accelerate the growth of cancer cells in the in vitro model as well as in vivo model. Since the HTK9-ME3, that histone mark, were upregulated in our early adaptive resistance state, we were interested about all the histone methyltransferases that are responsible for this histone mark, and we have found that there was a consistent upregulation of two histone methyltransferases, ZDB1 and ZDB2, across different cancer types in response to different stress, both at the mRNA level as well as at the protein level. So we thought that it will be a good target to prevent the formation of adaptive resistance. So we have done knockdown of that gene, and we have found that when we knock down ZDB1, ZDB2, it can decrease that histone marks, and we could actually sensitize those knockdown cells with the different chemotherapy or targeted therapy. So what we have observed in a nutshell from this part, there is a common adaptive mechanism in cancers, and this histone modification, which is one of the major type of epigenetic modifications, we have found that in our model they are generic for any kind of stress, and due to the global increase of the repressive histone mark HTK9-ME3 and decrease of active mark, it characterized the formation of RETCs, and silencing of ZDB1, ZDB2 induces drug sensitivity. As a follow-up, we have also published other papers as a follow-up studies where we have seen try to understand the adaptive resistance model, not only the models that we have developed in our lab, but also the in other labs, what they have published, and try to understand that what could be the common causes of adaptive resistance, and could that be due to the epigenetic modifications. And interestingly, I also did a side project where we have tried to target cancer cells with a plant-based extract, which is mainly found in the Asian region, um, called Magnanol. And we have found that this has the potential to target key pro-survival signaling pathway, PI3K pathway, and it can also downregulate some epigenetic modifiers, and then it can sensitize different forms of melanoma. And it was published as a cover article due to the interest of that compound, I guess. So the second part would be more relevant to the current topic, and I try to explore a bit more with the epigenetic inhibitors for untreatable melanoma, and how we can advance the epigenetic inhibitors for this untreatable melanoma. So when I started this lab as a postdoc, we published a nice review paper where we explore the epigenetic alteration in the context of melanoma, adaptive resistance, immunotherapy, and what could be the potential of epigenetic regulators to prevent adaptive resistance, as well as a alternative therapy to the current therapies that is available, or I should say that what are the standard care of treatment available. So briefly, what we are interested in the lab is to study the epigenetic regulators to understand what is the sex bias role in the patient. That means could it be like epigenetic regulators can lead to, or can give us some answer that why patients survive based on their gender. I'll come into that to the last part of my topic. And we also study because of whether we can induce anti-tumor immunity, because epigenetic inhibitors actually repress a lot of repetitive DNA, endogenous retrovirus, and melanoma antigens, which actually prevents anti-tumor immunity. So if we can re-express those by targeting those epigenetic inhibitors, epigenetic modifiers, we can actually induce more immunogenicity so that it can enhance the anti-tumor immunity. And there is a problem with T-cell exhaustion with immunotherapy. So with whether we can reverse the T-cell exhaustion, that is a current problem with the immunotherapy in the context of melanoma. And ultimately, whether we can develop the potential treatment option for the untreatable melanoma. So we try to explore what are the epigenetic inhibitors currently progressed in the clinical trials so far. And there has been considerable number of epigenetic inhibitors which has progressed or currently in the clinical trial, either alone or in combination with immunotherapy. So which suggests that it can be it can either complement the current therapies, or it can be a potential therapeutic option for the untreatable melanoma. So if we look at melanoma at a glance, so melanoma is the deadliest form of skin cancer. It is actually developed from the melanocytes. So melanocytes are the cells which produce melanin, and due to the sun exposure, with the exposure of ultraviolet ray of the from the sun, these pigment producing cells sometimes can contain mutation. So that means if you are overexposed to sun without any protection, without any sunscreen or without any protection, that means those pigment producing or melanin producing cells can harbor mutation in the cells, and that can lead to uh, excessive or abnormal growth of these melanocytes, which we call nevi, and that can ultimately progress to melanoma formation. And it initially forms the primary melanoma, and then it can migrate to lymph node, blood vessels, and it can ultimately metastasize to different organs of the body, and which is really devastating for the patient. So the incidence rate is highest in Australia, and no surprise, because the EV index is also highest in Australia compared to other part of the world. And majority of the melanoma actually occurs due to EV exposure. Although there has been a lot of development, as I said, with the targeted therapy or immunotherapy for melanoma patient, but at least 50% of the patient develop adaptive resistance, which suggests that there is a rapid need or immediate need for the development of new therapies. So what I have done, and I'm just discussing one part of the project in this lab, is try to understand these epigenetic regulators, which call bromodomains. It can regulate the gene expression, and it contains different family members: BRBD2, 3, 4, and T. So it actually binds to the acetylated histones regions, and that it can recruit other protein, CDK9, into the complex, which actually, by phosphorylating RNA polymerase, it can accelerate the mRNA formation, and that can cause the expression of several oncogenes like CMYC, BCL2, and MCL1. So our basic idea is that if we prevent both these BET proteins and CDK9 proteins, we can actually sensitize the cell more compared to single inhibitors. So what we have found that high expression of this BET protein, BRD4 and CDK9, is significantly associated with poor prognosis. And from the melanoma patient dataset, we have found that they are positively correlated, and they have a positive interactions to progress melanoma formation in the cancer patient, in the melanoma patient. So what we have found that BET and CDK9 inhibitors synergistically kill melanoma cells in a large panel of melanoma lines. As you can see here, we have 51. We have tested on 51 melanoma lines, and we have found that when we combine BET and CDK9 inhibitor, there was synergistic cell death compared to single inhibitor, as you can see here, also shown in this heat map, that it can kill a wide range of melanoma cells, which is not sensitized with the standard care of treatment, but it does not kill the normal cells like dermal fibroblast. We try to understand the mechanism, and we found that this BET and CDK9 protein actually, this inhibitor actually induce cell death through apoptosis compared to other forms of cell death like pyroptosis or necroptosis. And when we have treated the cells with either apoptosis inhibitor or compared to other inhibitor of pyroptosis or necroptosis, we could prevent such cell death upon treatment with PET and CDK9 inhibitor, which suggests that it could be through the apoptosis pathway. The cell death could be due to the apoptosis pathway. We have done some preliminary studies before we went to the mice model or in vivo model. So this is called three-dimensional spheroid culture, where we can generate whether we can mimic the tumor-like structure in an in vitro cell culture condition. And when I tried the BET and CDK9 inhibitor, we could see there are more cell death, which is represented by the red color, and green represent the live cells. And then we tested this BET and CDK9 inhibitor in the in vivo model, and we have seen that BET and CDK9 inhibitor synergistically regress the tumor growth compared to the single inhibitor. It also leads to the more survival of the mice compared to single inhibitors. To see the global changes of gene expression, we have done RNA-seq to understand what are the gene expression changes due to this BET and CDK9 inhibitor treatment. And we have found that BET and CDK9 inhibitors downregulate multiple oncogenic pathways like U2F, G2M, or MYC. And what we have seen that high expression of this E2F gene signature, G2M gene signature, or MYC gene signatures were associated with poor prognosis in a melanoma patient, which suggests us that this could be an important biomarker of which melanoma patients are likely to respond to BET and CDK9 inhibitor. So in a nutshell, from this study, you can say that BET and CDK9 combination synergistically induce cell death in vitro, in 2D model, 3D model, as well as in vivo. And we have found that it targets actually BCL2 family protein, and from the pathway enrichment analysis, we have found that several cell cycle oncogenic pathways are downregulated, and survival analysis, as I said, it revealed a biomarker of response of BET and CDK9 treatment in melanoma patient. So which actually gives us a hope that it will be, if we can do further research, it might lead to a potential clinical trial in the near future. So this has been featured in several newspapers, particularly in Australian Cancer Research Foundation. They have featured our article. And then there was another study I have done as a side, and where we try to understand that why we know that female patient actually survive better than male patient. That means the mortality in the male patient is definitely higher, not only in Australia, but also in USA and UK. And so we try to understand that what could be the likely mechanism or likely answer for this better survival of female patient. And then we try to understand the X-linked epigenetic regulators, and we have found that there is a epigenetic modifiers which is called KDM6A and ATRX, particularly KDM6A. We have found that high expression of this one was associated with better survival in female patient, and and then we further found that this high expression was associated with interferon-related pathway in the melanoma patient, which could particularly interferon gamma related pathway, and that is essential for the response of immunotherapy or target therapy. And in the in vitro model, we have found that there is a counterpart of this histone modifier, which is called EZS2, and we have found that we could sensitize the cancer cells or melanoma cells with EZS2 inhibitor, and that was also related to the upregulation of interferon-related pathway in the melanoma cells. So that has been published as well, and it has been featured in the Nisato's government press release. I'll quickly go into the. So that was all about my brief research that I have done so far. So there is a question arise that what could be the skill set required for research. So it really depends on your research project, I should say. So depending on your research project, the skill set should be developed or require. For example, the skill set for wet lab will be different from the dry labs, right? So for example, in wet lab, I would definitely pursue with all the basic molecular biology techniques that are available in your research, for example, or in the lab. For dry lab, the opportunities are abundant. That means for bioinformatics, if you're interested to pursue higher studies in bioinformatics, there are a lot of web-based tools. There are a lot of softwares you can explore, and if you are a biologist, for example, and then you want to do bioinformatics, I would highly recommend to get some basic knowledge of R programming, and that is R programming is designed for the biologist actually. So I think it will be helpful. So it really depends on your research projects, what you want to pursue in your higher studies. So first, to define that one is very important. For the publication tips, I would definitely suggest that we should work on our strength and interest. So first, we have to define what is our interest, right? So without knowing our interest, we cannot pursue a publication. We cannot pursue for publication. Don't go for publication just for the sake of publication, which in Bangladesh, I understand, and sometimes we have to do that for scholarship and everything, which is fine. But at the same time, you have to work on your strength, and then you have to know your interest. For example, if you want to do you want to develop your research career in the human disease context, it is better to do similar kind of research with the limited resource that is available in Bangladesh, for example. So, and if you want to do plant-based or agricultural-based or environmental biotechnology-related research, then it is better to do those similar kind of research with the limited resource that is available around you. And then the question comes that, or the second advice I would say is to reach out for every resource available around you, around the lab, either it is wet lab or dry lab, and it is always better to get advice from your supervisor, from your seniors, whoever is doing PhD in abroad. It is always good to seek advice, even from your fellow friends, your junior students. So that will come into in the next part, what is called collaboration. And I think finally, or the foremost thing is the idea generation. It should come first, actually, but we can discuss it at the last as well. For idea generation, so any kind of research first come from a novel idea, right? So for idea generation, the more you read, there is no other alternative. There is no other shortcut of idea generation other than reading relevant research articles from the plethora of previous papers. So it is better to read more and more to get a particular idea and create your own idea and work on that. Collaboration for me is like solving the puzzles. The puzzles obviously is the research question, and how we can solve the puzzles together. And for a collaboration, I think it is important to have mutual trust and respect to each other, and then we have to know each other's strengths, and then there is a saying that smart scientists think alike. So it's better to work with similar mindset people, and then you can develop similar kind of research, and then you work on and progress on that. So we have to first and foremost thing is that we have to be respectful, and we have to have trust to each other. I think that is the key for research collaboration. So I'd like to acknowledge my two lab that is from Brisbane, Professor Helmut Schreider's lab and Professor Peter Sarsis' lab in Sydney, and all the research collaborators in national as well as international, our funding bodies, and all the lab members, my past lab members and my current lab members. So with that, I'd like to thank you for your patience for listening to this seminar, and I'd like to have some questions. You have to unmute yourself.
Thank you, sir, for your precious space. Hope that it will be helpful all of the participants. Now I read out question from the comment box, and the one question come out from the গবেষক হতে চায় be research video. Thanks for your delivering very insightful lecture. Hope concerned learner will be benefited from it. Can you please tell how to start collaboration with renowned professor as a beginner?
As I said that your research should be aligned to whatever you are proposing, like for example, renowned professor, you want to do a collaboration, so your research has to be aligned, and then you just tell them that our research is aligned and I'm particularly interested to do this kind of research. And if you know their strength, and then you can also tell them that this is my strength and this is what I'm proposing. I think people are happy to do a collaboration.
Without hesitation, can you directly question in the comment box? So we know that cancer is a complex disease. Its mechanism is really maximum to unknown. So
yes, it is. I mean, the main thing is that we, like scientists around the world, are trying to understand why some patients respond to a particular therapy, some patients do not respond to a particular therapy. So that is always challenging. So we're trying to understand that why some people do not respond to a particular therapy, or even if they respond to a particular therapy, why do they develop resistance in the later in the long term. So, and then there's a question arise that whether we can develop alternative therapy, and then we are proposing that epigenetic therapy could be a potential alternative therapeutic option for the patient that are not responding to current therapies.
So we already know that chemotherapy, radiotherapy has contained a side effect. So epigenetic therapy is alternative treatment. So many patients will recover very soon. I hope that scientists, researchers quickly discovered epigenetic therapy for valuable patients who are uh, already uh, affected different kind of cancer with other disease. And actually, it's a biomarker identification is a very recent topic to cancer with the other disease. And if we early stage, if we detect biomarker in early stage of cancer, so we easily can, the mortality rate of cancer may be reduced if we early detection of biomarker. So how what types of biomarker can we detect it in your lab experience, sir? Can you explain it?
So scientists around the world, tirelessly working on the biomarker of any cancer types, and it depends really on the type of cancer. For solid cancer, it varies different from even from the solid cancer. If you look at lung cancer, the biomarker could be different. Um, um, for example, prostate cancer or melanoma biomarker could be different. If we look into the blood-related cancer, the biomarker could be different. So people look for the different oncogenes, which causes cancer formation. And from the advent of whole genome sequencing, we now know that what are the percentage of mutation in a particular gene. For example, for a particular cancer types, this particular gene A, contains 30% or 40% of mutation. So that could be a biomarker. For example, if the gene is one of the top 20 or top 10 genes that is highly expressed in the cancer patient compared to the normal, so that could be a biomarker. So people are looking in various different contexts.
The second question is whether the gene treatment against cancer cell is limited to this specific cell or not.
Okay, yeah, that's a very good question. So gene treatment, so we're not doing gene treatment. So gene therapy is kind of different from what we are doing. Gene therapy is definitely repairing a gene in a particular cell. So that's why it is kind of specific. So for this kind of epigenetic therapy, so epigenetic therapy is also targeted therapy. Is specific for a cancer cells. It is meant to be specific for cancer cells, and which should actually target cancer cells specifically and without altering or without damaging or without providing any harmful effect to the healthy cells.
Sir, I have one question in bioinformatics approach. Uh, we can usually recently identified microarray data analysis or RNA sequence data analysis, and we then identify most significant hub genes by protein-protein interaction. Then we selected one or two protein, and then we know that recently most phytohistogenic compounds are found in different plant source, and we then identify a protein and with docking purpose with phytogenic compounds. And we found a strong score, docking score for each phytogenetic compound with a selected hub gene or hub proteins, and it's a very valuable approach. And I hope uh, this area is a huge purpose with bioinformatics. And if you can suggest who are new scientists can research in this area, so what is the application of this and how it is possible to publication this type of research? Can you explain, sir?
Yes, I mean, depend on the research question that you are trying to address. For example, molecular docking is getting a lot of attentions nowadays, but not only with molecular docking, but any kind of bioinformatics research demands further exploration with wet lab experiment. But it is a very good start point with the resource that as I have mentioned in my lecture, that with the resource that we have, if we can just explore and then tell that this could be a potential target. For example, if you identified with all the gene expression and microdata, you identified a particular protein or particular gene family that is important as a for a particular type of cancer, for example, and then you see that it also has a role in survival. So I would definitely see what is the role in survival of that particular cancer types. And with the in silico analysis, if you see that there are some docking compounds available for targeting that protein, which will be definitely helpful. So in a bioinformatics paper, we provide insight, which always tell that it requires further experimental validation, but it is definitely a good starting point.
Thank you, sir. Uh, Thank you both for joining our today's program, and a special thank Dr. Abdullah Imran sir for his valuable time with sharing his experience, lab work experience especially, and suggestion for new researcher.
Thank you very much for having me here. I mean, my pleasure to present before you. After a long time, I think I was presenting in terms of a BD community or to my students or whatever the fellow researcher around there. So it is always pleasure to talk to my own research community, and it is ensuring that. And I'm really thrilled to see, or really happy to see that people are getting interested about research, not only in the biomedical science, but also in other areas. And I hope that you should just keep the spirit up and then just work on your strength and don't dream small, dream big, and definitely you can achieve whatever you want to achieve in your life in terms of research.
Thanks again, sir, for your valuable time. Also, thanks to the audience for your participation. Announcement: our next research idea series will be held very soon. To get update of our activities and follow our page, join our group and subscribe to our YouTube channel. We are happy to inform you that we are now a family of more than 23,000 members, including group, page, and channel within 9 months only. Stay with Gabeshak Hote Chai, be researcher BD, and invite your research enthusiast friends to our platform. Wish you all success and good health. Allah Hafiz.